we're going to get a little more in depth into exactly what photosynthesis is now. We kind of have in our mind that photosynthesis is a reaction. And in the past we've said that plants do photosynthesis to make their own food. Plants are producers. They're going to make their own food, but really all that they're trying to do is they're trying to make an organic compound. Now we know that an organic compound has C, H, and O, and so we have to find some way to take carbon dioxide and water and build an organic compound like a sugar. So the idea of photosynthesis is that plants and producers are able to take light energy and transform it into chemical energy in the form of glucose, C6H12O6. And we know that glucose might be then used to build the cell wall, make cellulose, might be used to make starch, might be made to make a sugar that would be a nectar to attract bees to help reproduce. So what we want to keep in mind is, what is the purpose of photosynthesis? We're trying to build an organic compound. Take two inorganic compounds that are in the environment and build an organic compound. If we're forming bonds, that's going to take energy. And so we use energy from the sun in the form of light energy. And so we then talked about what is light energy and the idea that not all energy is equal, that we have some that is higher in energy, some that is lower in energy. We've got this whole spectrum of energy. And so then we looked at, well, what is the structure that plants have to help them absorb light? And so the structure is called the chloroplast. This is the double membrane bound organelle found in some plant cells, the ones that get light. And inside of the chloroplast are these thylakoid membranes that are stacked in what are called granum. And in those membranes are pigments. These are going to be proteins that are going to be embedded in there to help absorb light. And we've got what we call an action spectrum. So a whole spectrum of different pigments that work together to capture a whole lot of light. Some plants are actually able to capture ultraviolet, some infrared, but most of it's here in this Roy G. Biv. But noticing that green is kind of missing here, that most plants cannot absorb green light and are therefore going to reflect green light, and that's why plants look green, because they're not using the green light. And so that's kind of where we ended, with this idea that plants are going to absorb light. But what are they going to convert light into? And so this is where we're going to look at what's called the light-dependent reactions. Photosynthesis is divided up into two main stages. And so when we first look at the light-dependent reactions, we see light coming in. And this light is going to be absorbed at many different proteins, but it's going to be funneled or centered to a single type of protein, and that's going to be able to absorb light at the wavelength P680. So we're going to see there are actually two different what we call photosystems, or two different types of ways to absorb light. Photosystem 1 is really able to absorb best at 680 nanometers. And this is actually going to be chlorophyll A. Chlorophyll A is the main pigment here in this first photosystem, or organization of pigments. And sadly, it's called photosystem 2 because it was discovered second. It is the beginning of this reaction. So the very first input of light is going to be absorbed at P680, and it's going to excite some electrons. Sound familiar? We know that electrons are a main source of chemical energy. So we're going to use this light to excite the electrons. Where are the electrons coming from? The electrons are actually coming from the splitting of water. we know that water is used in photosynthesis. It's actually used at the very beginning. And we're going to split water, we're going to release oxygen, we're going to release hydrogen ions, but we're going to use those electrons to capture that light energy. So we're putting the energy of the light into the energy of these electrons. The electrons are then passed to some other proteins. Sound familiar? Another electron transport chain. As we pass the electrons, we're actually going to use their energy to pump hydrogen ions. Sound familiar? One thing we do have to keep in mind here is that we're talking about being on the inside of the thylakoid membrane. So this time we're pumping hydrogen ions into the internal space between the membranes. We've used some of the energy, so we actually take a second input of light. This time it's the pigment chlorophyll B. So a different version of chlorophyll is able to absorb a different wavelength, P700, and excite the electron again. So we initially excited the electrons, but then we use some of that energy to pump hydrogen ions. So we excite the electron again, and then we pass it to an enzyme that will pass the electron to a carrier. This time our carrier, the oxidized form is NADP+, and the reduced form is NADPH. And this is actually called the Z scheme. Because we excite, we use the energy, and we excite again, so you can kind of see that Z shape. And what we've done, of course, is create a gradient so that we can do chemiosmosis. We'll actually make ATP again. So a slightly different view of this reaction is happening here. It's a little bit more set up like we're used to seeing in the mitochondria. Keeping in mind, this time we're in the chloroplast. These are the stacks of thylakoid membranes. And we see a different diagram that has the basic same idea. We see light coming into photosystem 2. It doesn't show the Z scheme as clearly, so we almost wish we could merge these two pictures. But we do see all the important reactants. We see light coming in to photosystem 2, the splitting of water, releasing oxygen, creating a hydrogen ion gradient. This time the hydrogen ion gradients are here in the thylakoid space. The energy from the light allows us to pump more hydrogen ions in. We get a second input of light and excite the electron again to now pass it to our electron carrier. So there's kind of our electron transport chain. So the nice thing is ATP synthase isn't shown there. We know that ATP synthase isn't receiving any electrons. The electrons are being transported here to pump the hydrogen ions and to be passed to this electron carrier. Our gradient of hydrogen ions means that we can make ATP. And so the light-dependent reactions, these are called the light-dependent reactions because they depend on light. Light is a limiting factor. If I don't have light, none of this will happen. If I don't have water, none of this will happen. If I don't have an oxidized electron carrier, none of this will happen. If I don't have hydrogen ions, if I don't have the ATP synthase enzyme, none of this will happen. And the idea is that at the end of the light-dependent reaction, I have ATP and NADPH, both of which are really sources of energy. This is going to be electron energy, and this we know is kind of the idea of the phosphorylation energy. That idea that when I add that phosphate group on, it could change the shape of something. So we'll see the use of ATP and this electron carrier later. So it very much is like photosynthesis kind of in reverse. We see here that instead of pumping hydrogen ions out, we're actually pumping hydrogen ions in. So the electron transport chain has a similar idea, but this time we're taking the energy in from light. out that there's a really important factor that has to happen in terms of regulation. And this is going to be called cyclic photophosphorylation. So the creation of ATP here is actually called photophosphorylation. Photophosphorylation. During respiration, it was called oxidative phosphorylation. Now we call it photophosphorylation. So it turns out that we actually need more ATP than we do NADPH. So we have a higher need for ATP. So we're actually going to try to make more ATP than we do NADPH. And so what this means is that sometimes we don't pass the electron on quite yet. Sometimes we use that electron a couple of times before we pass it on. And that's called cyclic photophosphorylation. It's a way to control the speed of these reactions. And so what will happen is kind of the second part will happen over and over again. We'll use light energy, and that's not shown here. We'll use that second light energy to excite it, and instead of passing it on, we pass it back, and we pump more hydrogen ions. Hydrogen ions get pumped. So cyclic photophosphorylation is the idea that photosystem 1 can actually pass the electron back to continuously pump hydrogen ions until we've reached the need that we have for ATP, and then we'll pass the electron on to NADP+, so that it can become reduced and become NADPH. So the idea of the first part of photosynthesis is that we're using light, and we're putting the light into these chemical bonds. We're using electrons to store the energy into chemical bonds. And so the next part of the notes is really just kind of a summary. Where does the energy come from? Where does the electrons come from? Where did the water come from? Where did the oxygen come from? Where does the oxygen go now? Where did the hydrogen ions come from? Where did the ATP come from? What will the ATP be used for? And so this is kind of an important idea. We're going to use these to do what we call carbon fixation. This is when we're going to be building the organic molecule. So ATP and NADPH will actually go on to the next part of photosynthesis called the Calvin cycle, and that's where we actually take CO2 from an inorganic form to an organic macromolecule. And that is the light-dependent reaction. So really just taking some time to be able to talk your way through the electron transport chain and the creation of ATP and this important point of regulation at cyclic photophosphorylation.